right, in chapter 13, we are going to learn about how microbes and humans interact and how they are able to cause disease. So first of all, each human body actually has trillions of microbes that stably associate with them. That's that microbiome, referred to as multiple different things. Resident microbiota, also called the human microbiome, the normal flora. There are multiple kind of words for this, but essentially it's all of the microbes that typically and constantly live in or on our body. Again, the main key is that most of them are beneficial, but they all have an impact. And so the term holobionte is the term for the human plus all of its resident microbiota. So human cells plus all of the microbial cells. So we've talked about this, especially with antibiotics, and how whenever we take antibiotics, it disrupts those resident microbiota and can cause problems. But again, we have human cells, we have a lot of microbial cells as well. So a few terms then to go along with this that we use a lot, but just to quickly define them. So infection, this is where there's a pathogenic disease-causing microorganism that has been able to penetrate the host defenses and multiply. So that's kind of the key. So just being in or on a person doesn't necessarily indicate an infection. They have to be able to multiply, and that's where our host defenses try to stop that. Then following that up with disease. So this is a pathological state that results from the cumulative effects of that infection. So where there's disruption to the tissues and the organs after those microbes have been able to multiply. So just the multiplication of a pathogenic organism is an infection. When they actually cause significant harm to the host, to the tissues or organs, that's whenever we have the disease. So deviation from health. And then an infectious disease, to put those two together, this is where we have disruption of tissues that is caused by microbes. So there are multiple diseases like heart disease. They're not caused by a microbe. So just to differentiate, if it's a disease caused from a microbe, it's called an infectious disease. All right, so a little bit more about, again, that normal flora, the normal biota. Again, this is a large, diverse collection of various microbes, mainly bacteria, but also some fungi and some others can be there as well. Again, just lots of different terms that we kind of have for that. So just understanding, again, it can be called a lot of different things, but it all means the same thing. The microbes that normally live on or in our body. And again, it's a lot of different things. So bacteria, fungi, even viruses or protists. But again, they have a profound effect. Again, most of them beneficial. They help with digestion. They help protect against pathogenic organisms from being able to take hold and multiply. So we just kind of look at how all of this works together. So the word colonize just means that something is on and multiplying on the body. So we are colonized by lots of resident microbes that are, beneficial. There are some that are potentially pathogenic. So when we shake somebody's hands or somebody sneezes, or we just come into contact with some pathogenic. And then we also have some transient microbes that are only there for a small amount of time. So that's why we wash our hands, is to get rid of those potential pathogens and just transient microbes that don't typically reside in or on the body. So again, of all of these microbes that are on here, again, they're both potential pathogens, and the transients can simply exit the body. They're not, again, it's not part of our normal microbiome. They're not permanent fixtures, so they can be gone, so to speak. Now, occasionally those potential pathogens can cause infection, and sometimes that can even be the resident microbiota. And again, that's the problem with those super infections. Whenever we take antibiotics, it can let some of those resident microbiota that are kind of kept in check, can allow them to cause an infection. And again, infection just means that they are able to multiply above and beyond their normal capability. Now, most of these are going to be asymptomatic, and they're cleared, and our immune system is going to get rid of them. Unfortunately, some of them can actually become to that disease state. So if we have some here that are in that infectious state, again, some of them can reach the level of the disease where they can harm the tissues. Ultimately, once that disease occurs, unfortunately, of course, it can lead to death. Our immune system can eliminate them and destroy them. The other thing, though, is even some of those pathogenic bacteria that we come into contact with, some excuse me, some of them may actually become part of that normal flora, in which they become part of, again, that term holobiont. So again, once we have an infection, we have microbes multiplying. Again, the options are getting rid of them. It can cause disease. It could ultimately lead to death, or they could even start becoming part of that normal flora or those resident biota. Now, again, as we mentioned, this normal biota is beneficial. So some of those benefits, even early on, they can help development of the organs. They prevent, though, overgrowth of harmful microorganisms, so those pathogens. If our body's covered with good bacteria, it's harder for the pathogenic ones to attach. And if they can't attach, then they can't multiply. So we call that microbial antagonist. Essentially, it's the effect of the good microbes against the intruders. So it's good bacteria fighting off the bad bacteria. So the microbes, again, are a steady, established relationship, then they're unlikely to come off. And again, that just covers the surface so that those pathogenic or more harmful intruders can't attach or multiply. Now, here are some different factors that can decrease our host defenses and actually make us more susceptible to infections. So the age, both spectrum, so the very young, so infants and the very old, both have weaker immune systems. Younger, they haven't fully developed. Older, they're starting to kind of wear out. So either of those can make us more susceptible to an infection. Various genetics, especially in the immune system, or it can be acquired. So conditions like AIDS, where it destroys or weakens the immune system. Pregnancy can decrease the immune system. You're more susceptible during pregnancy. Surgery and organ transplants, because they're given immunosuppressants so that they don't reject the organs. There can be different underlying diseases. So cancer, just because the body is kind of busy trying to fight off the cancer, so it's less able to fight off pathogens. Altering the liver, diabetes is another one. So again, it just alters the immune system that makes other infections more likely. Chemotherapy and immunosuppressive drugs, and not just for transplants, but for other reasons, again, cancer, or again, other diseases. Stress, whether it's physical or mental stress, again, just weakens that immune system, and the presence of other infections. Now, there are some different kind of routes in which infections occur. So some of them are endogenous infections. So this is caused by that biota that's already in the body. So it's not something new that we've come into contact with, but this is when that normal biota is introduced to a new site that was sterile. So E. coli is a good example of this too. So E. coli normally is in our gut, which is okay. It doesn't cause an infection, but if it gets into the urinary tract, then it can cause a UTI, urinary tract infection. So whenever it's not in its normal spot, then it can cause problems. Another example here, Streptococcus pneumonia is normally in our normal biota, but in AIDS patients where they have a weakened immune system, it can actually lead to pneumococcal pneumonia. And that's just because they're not able to kind of keep it in check. So where does our normal biota start? So there's a growing number of doctors and scientists that believe that the fetus, so actually still in the womb, are seated in utero, so that they're exposed to different bacteria while they're still within the mom. And then after that, there's definitely exposure during birth. So as they're coming through the vaginal tract during a natural delivery. So there are a few studies about the difference between a cesarean delivery and a vaginal delivery, with the difference in the normal biota, because those with cesarean don't travel through, of course, the vagina. So then they have a different normal biota. And then breast milk also has lots of different bacteria that help set up that normal biota, because again, the babies actually can't quite digest food, so that normal biota actually helps them break down the sugars. So again, that breast milk may be necessary for having that healthy gut biome. So it's one of the advantages to breast milk over formula is that exposure to good bacteria that can then help with digestion. Now, a lot of formulas contain some probiotics. They have bacterial colonies that are in them to help establish that gut biome. So again, just kind of a visual here of where that microbiome comes from. So again, while it was previously thought that the fetus was sterile, it's now more increasing information that there is some transfer of bacteria from the mom to the fetus during development, definitely during birth. So of course, coming through the vaginal tract, but even during the C-section, they're still going to be exposed to some of those microbes as they're coming out. Again, breast milk has its own microbiome, but again, sterilized baby formula. So again, a lot of formulas now have cultures that are added, and then just the people that they come into contact early on, especially, as well as just their environment, things that they're touching or things that they're around, because again, there are microbes everywhere. So that helps set up that normal biota. All right, now how are these microbes actually able to cause an infection? So this progress of an infection. Again, a couple of terms, we've talked about pathogens before, but they are parasitic. They damage the host and can cause infection and disease. Now we also have the term true pathogens. So these are capable of causing disease in a healthy person. So that has a normal human or normal immune system. Whereas opportunistic pathogens, they can only cause disease if the immune system is compromised. So again, if there's one of those pre-existing conditions like cancer or immunosuppressant drugs or something. So a true pathogen can cause a disease regardless of the state of the immune system. Opportunistic, again, they take advantage of that weakened immune system. That's the only time they can cause an infection. A few terms here, because you'll see these in the healthcare system. So just to briefly mention them. So biosafety levels, this was adopted by the CDC, so the Center of Disease Control and Prevention. So there are 4 levels, and this refers to their degree of pathogenicity. So a level 1 organism has a low chance of infection, so it's not very harmful, whereas a level 4 would be one that's very virulent and has an extreme risk, so they can cause severe damage to the host, so very pathogenic. So again, level 1, pretty good. Level 4, not so good. So this term virulence that we just used, so virulence is just talking about the degree of pathogenicity. So it indicates the ability of the microbe to, first of all, establish itself. So to attach, does it have a capsule? Does it have fimbriae or pili or something? And then once it's attached, its ability to cause damage. Does it release a toxin? Does it cause a large immune response that can devastate the person? So again, essentially how bad the bacteria is or the microbe is its virulence. And then we have virulence factors. Those are the characteristics, like I just mentioned, again, a toxin or a capsule. Those are factors that allow the microbe to become a pathogen. So they're referred to as virulence factors. And then also infectious dose or ID. This is the minimum number of a microbe required for an infection to occur. So, for example, if you eat potato salad that's been setting out for, say, 10 minutes, it may have a little bit of salmonella that's in it just from the food, just producing it with the eggs. So let's say it has 10 Salmonella bacteria in it. So that's a pretty small dose. So your immune system, just traveling through the gut and the acidic nature of your stomach, that's probably going to destroy them, and that's not going to be enough to cause a problem. But now if that potato salad's been sitting out for a couple hours, and now there are 1,000 or 5,000 salmonella bacteria existing in that potato salad, now it's less likely that all of those are going to be destroyed by the stomach, and some of them will be able to set up an infection. So that's what we're talking about with an infectious dose. So the smaller infection equals greater virulence. So again, a very pathogenic, a very virulent microbe doesn't take as many. So the infectious dose would be maybe only 100 that you would be exposed to, and it would cause disease. Whereas other microbes that are less virulent, it would take like 1,000 or 2,000, so that infectious dose would be higher. So if the infectious dose is really low, that indicates that it's very virulent. All right, so there are three main variables with this host relationship. So first of all, the host genetics. So everybody's different with their genetics. So we have different responses to the same microorganism. And this is just due to our genetic variation. So we all have different antibodies. So poison ivy might be a good example. Some people can roll around in poison ivy and nothing happened to them. Others could roll around and get a small infection, and others would have a severe reaction. Again, it's the same thing, but there's genetic variability that causes different infections. It's the same way with microbes. And then also just the exposure to a particular microbe. So again, is it a very virulent microbe? Is it not so virulent? And then again, the overall health of the host. Are they immunocompromised or are they healthy? So here are the steps that actually lead to a disease. So this is kind of similar to what we looked at with viruses. So first of all, they have to get in to the tissues. So again, we have a lot of kind of natural defenses like our skin. So is there though a puncture? Do we have a wound to the skin? The GI tract, of course, things that we eat or consume, things that we inhale through the respiratory, things in our urogenital, and just those endogenous biota that were there to begin with. So how is the microbe getting in to the body? Then once they get in, that's still not enough. They could come in and just leave right away. So next they have to attach. And those are things like virulence factors, again, the fibrae, capsules, surface proteins, or if we're talking about viruses, those spikes. So once they get in, they now have to attach. So that's the role of the normal biota. It helps cover up our surfaces so that those pathogenic bacteria can attach. But if they're able to kind of outcompete the normal biota, or they're able to get in and attach, now what they have to do is survive the host defenses. So once they start attaching, our immune system should recognize them and try to get rid of them. So in order to cause disease, the microbe may be able to avoid phagocytosis or being eaten by our immune system. They can essentially hide inside of the phagocyte, or they can just not cause a reaction. They can look like something that belongs there and not activate the immune system. So if they've gotten in, they've attached, and our immune system hasn't caught them, now they're able to replicate. And so now they have to have a way to cause damage. And that's where those virulence factors come into play again. So it may be direct damage from enzymes or toxins. It could be from an excessive host response. So it overactivates our immune system, which leads to inflammation and fever as our body's natural way of getting rid of it. But sometimes the immune response is as bad or worse than whatever we're exposed to. And it can cause epigenetic changes, so it can actually change our DNA. So we talked about like viruses, they can add oncogenes or genes that cause cancer due to the presence of that virus. And then finally, exiting the host. So some of the portals of exit, just as it gets in, it gets out. So essentially any opening. So respiratory, again, sneezing, coughing through the saliva, sloughing off through the skin, from the GI system coming out through the feces, or the urogenital tract, or also through our blood. All right, so just going through each of these steps in a bit more detail. So again, step one, getting in. So this is the route taken to initiate that infection, usually through the skin or some mucous membrane, like our respiratory system. So the source of where that agent comes from is either from outside, so exogenous, it's coming from outside of the body, the environment, against somebody sneezing or somebody touching us, or it can also be endogenous, and so that's already on or in the body. So from the normal biota, or something that was just previously kind of silent and not a problem. So the portals of entry include the skin. So they get in through nicks or abrasions. So that intact skin is a great barrier. So whenever we go through the immune system, we'll talk about kind of innate or inherent things that we have, and the skin is one of those. It just prevents things from coming in. Through the GI system, so anything we eat or drink or just ingest. So again, we have some natural defenses, those digestive enzymes, as well as the acidic nature of our stomach. Those are kind of natural defenses as well. The respiratory system, either through our mouth or our nose, so breathing into our lungs, or through the nasal cavity, allows microbes in. And then urogenital, so think about like sexually transmitted infections, but also just coming across the skin or mucus of the penis, the external genitalia, vagina, cervix, or urethra. So again, coming, of course, through sexually transmitted, but also some, again, we think about, especially for females, whenever they wipe, going front to back. Otherwise, it exposes E. coli, can introduce it into the vaginal tract. So again, that's through that urogenital entry point. So now, once it's gotten in, again, attaching. So there has to be an adhesion, a way for the microbe to gain that stable foothold so it can attach. So this is dependent on binding between specific molecules. So just like viruses, bacteria and other microbes have to be able to attach to a cell. So sometimes, again, microbes can cause disease in one species but not another, because of those specific molecules or receptors that are different. So again, it's limited only to the cells that they can bind to. So if they can't bind, they can't cause an infection. Now, once they're attached, then it can invade the body compartments. And then another term here that kind of goes along, quorum sensing. This is chemical communication between nearby bacteria that can help set up that infection. So essentially, like, hey, I found a way to get in, and they bring all their buddies to the party. So kind of like ants almost, and how they find a food source. It's kind of the same thing with bacteria. They can release chemicals that invite other members essentially to their infection party. All right, so just kind of looking at the adhesion. So we've talked about these parts before. Again, these would be virulence factors. They're things that allow them to cause an infection. So the fimbriae, again, the projections, appendages that stick out that they could attach to the host cell, but it has to be something kind of specific. The capsule, that's that sugary layer on the outside here, so it's just kind of sticky and lets them attach. Viruses have the spikes that come out of that capsid that allow them, again, to attach to specific receptors. So if the host or that cell doesn't have the receptor, the spike can't attach. All right, now once we've attached, now we need to survive the host defense. So we talked about phagocytosis, but a phagocyte is a white blood cell, part of our immune system, that engulfs, essentially eats a pathogen, and the purpose is to eat it to destroy it. So it breaks it down with different enzymes and chemicals inside of the cell. So it's a way of removing the pathogen from the tissue or from the blood and breaking it down with those enzymes. However, different pathogens, again, virulence factors that allow them to prevent phagocytosis. So one is that they can avoid phagocytes. So the capsule is a really good one. So because it's covered in that kind of sugary layer, our immune system doesn't recognize it. It can't bind to it. So those phagocytes don't engulf it. Or they invade some part of that process. So they're resistant to the enzymes that the phagocyte is using or to those chemicals. So it gets engulfed, but it doesn't get destroyed. Then again, causing that disease, again, different virulence factors. So structures, products, capabilities that let it establish that infection. So it can damage the host, whether it's producing a toxin, overactivating the immune system, but something that alters the tissues or organs to cause that. So the damage that it's doing to the host, so there are a few different ways, kind of three main ways here that they cause damage. One, secreting proteins. Again, toxins are good examples, but also enzymes. So there are enzymes that break down blood cells or break down tissues. That would be an example. Again, overreaction of the defense. So it's not so much the pathogen causing the disease, but it's actually the immune system. So again, those phagocytes are coming in, but they overactivate the immune system and it causes a lot of damage due to the immune system, more so than directly from the pathogen. And then the other way is that they can alter the DNA through the genome or transcription. So it can alter the proteins that are being made. It can change the DNA. So those are referred to as epigenetic changes. So for example, again, that oncogene being inserted by a virus, now it's producing a cancerous something that's overactivating cell division that leads to tumors and ultimately to cancer. Now, a little bit more here about the enzymes. Again, there are enzymes and toxins, but just a little bit more about them. So first of all, exoenzymes, these are ones that are secreted. So they're made inside of the bacteria, fungi, protists, worms, and then released. And those enzymes, once released from the pathogen, they break down or inflict damage. So they can dissolve barriers from the host cell, like the skin or the mucous membrane, and that promotes the spread to deeper tissues. So for example, there's mucinase, which breaks down mucus, keratinase, so the keratin in our skin. So if it releases these enzymes, it weakens the skin and allows them in. Hyaluronidase, kind of more around the cartilage in the bone. So again, these enzymes help it be able to get in and cause damage. Now, the toxins that can get released, again, a toxin is just a chemical that is essentially poisonous to other organisms. And they can either, and here, whenever we're talking about virulence factors, we're talking about exotoxins. So these are secreted, just like exoenzymes. Exotoxins are secreted by the cell. There are lots of different types. So like tetanus is due to a toxin released from the organism, multiple GI problems. Again, it's the bacteria releasing a toxin that is damaging to the host. Now, just to come back to this, endotoxin is different. So endotoxin is not secreted. This is the lipopolysaccharide that is only on the gram-negative bacteria. It's on that outer membrane of gram-negative bacteria. So this is called endotoxin because it's not released, it's not secreted, but it is toxic to our cells just by the presence of a gram-negative bacteria. That lipopolysaccharide, again, that gets released from the outer membrane, has its own toxic effects. So just kind of looking at these two, differentiating between exo and endotoxins. So exotoxins, they're toxic within minutes, whereas endotoxins, we need a high dose. So lots of that being released and kind of broken down from those gram-negative outer membranes. Exotoxins are specific to a cell type. So again, it's like hyaluronidase or keratinase. It's breaking down specific molecules. Whereas endotoxin is kind of systemic. It's just the presence of that activates an immune response that's kind of non-specific. Exotoxins are small proteins. Again, endo, there's only one. It's lipopolysaccharide that's in the outer membrane of the cell wall of gram-negative bacteria. Exotoxins are not very stable, so they're a little easier to break down, whereas the endotoxin, that LPS, is very stable. Exotoxins can be converted to toxoids, and a toxoid is just an inactivated toxin used by a vaccine, so they can be destabilized, and it's a way of training the immune system against it, so it can actually be used to help make vaccines. LPS, endotoxin, cannot be inactivated to be used as a potential vaccine. Exotoxins stimulate antitoxins, and this is essentially an antibody that reacts to a specific toxin. So we make antibodies that recognize a cell. Antibodies also recognize different toxins. That is not the case with endotoxin, so we don't produce antibodies that recognize the LPS or that endotoxin. Exotoxins usually don't create a fever or lead to a fever, whereas endotoxin, that LPS of gram-negatives, do cause fevers. Again, exotoxins are secreted from those. However, endotoxin is released from the cell whenever it's destroyed. So whenever a gram-negative cell is intact, LPS doesn't cause any problems. So our normal biota has lots of gram-negative bacteria, so there's endotoxin within that. The problem is whenever the cell breaks apart. So as we see here, whenever that gram-negative cell is destroyed, it's lysed or something, now as it's broken down, that LPS in that outer membrane, now it gets released. That's when it's a problem. And then exotoxin can be made by different types of cells, whereas again, endo is only in the gram-negative.